All right, so we're working on biology notes here for section 5 of chapter 6 on traits and probability. So your objectives in this section are to be able to describe monohybrid and dihybrid crosses, right? We're going to do some Punnett squares to keep track of traits in these crosses, and you need to know how to do that. And then we're also going to talk about how heredity can be illustrated mathematically. So first off, we need to do a couple crosses. We're going to do monohybrid crosses first, which means we're only keeping track of one trait at a time. So I need to cross parents of different genotypes. In this first example here, I'm crossing purebred purple with purebred white. So it's homozygous dominant for the purple and it's homozygous recessive for the white parent. And I want to cross those and see what kind of results I'm going to get. So in order to do one of these things, you need to make a square and then divide it into 4 boxes. And I'm going to put one parent on one side, so homozygous dominant here, and then I'm going to put the other parent on the other side, and then I just fill through to see what results I get. So I always get one allele from each parent. For that first square, I'm getting a capital and a lowercase, one from each parent, and I just fill in just like that. I get a capital P for both of these first 2 boxes. I get a lowercase p filled through for both of those boxes. So you just fill out the whole thing from the top, from the side, and it gives you all your different possible combinations. Now in this case, it's pretty boring because I get a bunch of the same thing. All of those offspring are the same. They're all going to have one dominant and one recessive allele, so they're all going to be that purple color. And in Mendel's experiment that we talked about before, remember all of that first filial generation were purple. So that's exactly what we would expect. It's that second generation that was surprising, that had the little twist to it. So that's this example over here. If I'm going to cross 2 purple heterozygous individuals, I should see some different results. So again, make your square, put one parent on one side, the other parent on the other. Doesn't really matter which one goes where. In this case, they're both the same, so it definitely doesn't matter. But again, fill down from each side, fill across, and I get these different combinations. So this time, I should see, at least from that phenotypic standpoint, I have that 3-to-1 ratio that Mendel saw in his 3rd generation, that second filial generation. So there's 3 purple and 1 white, but there were 2 different ways that you could be purple. You could either be homozygous dominant, like this first one here, or you could be heterozygous, like the other 2. Maybe I shouldn't put that white mark there. Maybe I should put it in here, because those actually are carrying that white trait, right? But all 3 of those are going to be purple. At the end of the day, that purple masks the white. So that's why we see that pattern. That's what's underlying in those genotypes. That's what Mendel had to figure out working backwards. So for us, it's a little bit easier if we know that we have 2 heterozygous parents. You're always going to end up seeing that 3-to-1 ratio, dominant to recessive. Okay, so as long as it's just a monohybrid cross, as long as we're only dealing with one trait, these are fairly simple, fairly easy to do, but you need to be able to interpret your results when it's all said and done. So a test cross. Remember in that 3rd generation, I had some purples that were homozygous dominant and some that were heterozygous. Mendel can do a test cross now to decide whether that offspring is homozygous or heterozygous, right? The only thing we did know about the genotype was that there was at least one capital P. If you see purple, that has to be true. But I don't necessarily know what the second letter is going to be. So that's why I would perform a test cross. And in a test cross, you're going to cross that organism with the unknown genotype with an organism with the recessive phenotype, right? So in this case, that would be the white flowers. So I crossed one of those purple flowers from the third generation with a white flower, and I can find out what it is at the end of the day. So really there's only two options, right? It was either going to be homozygous dominant or it was heterozygous, capital and lowercase. So I can just do two different crosses to see what results I might see at the end of the day. And then if I do an actual cross with the actual flowers themselves, purple and white, I should be able to find out what it is. So both I'm crossing with that homozygous recessive, a white flower. So I just go ahead and fill through and see what I should be expecting at the end of the day. If it happens to be homozygous dominant and I cross it with that white flower, all of the offspring are going to end up being purple. If it's heterozygous and I fill through, only half of them are going to be purple. The other half are going to be white. So down at the bottom there it says if the results of the test cross had one purple to one white, it was a 1-to-1 phenotypic ratio, what would the actual genotype of the purple parent be? Since in only one of my two test crosses up here did I even get any white offspring, I know automatically that this is my genotype, that my unknown allele up here was that lowercase p, right? That purple must have been heterozygous if there were any white offspring at all. So it's just kind of a logic question. Think about what the possibilities are and then what you actually see from experimental results. If you did get white, it had to be heterozygous.